Hi, everybody. Welcome to part two. We're going to go over some more details that you need, warned you, chemistry, of all the molecules that are important for us to talk about nutrition. My wife, one aside, my wife saw the last video and said that my lockdown beard and usual shadow are not very attractive. So I promise next time I'll shave. I think she's also talking about trimming my hair, but we shall see. So if you recall, the last time we chatted, I left you with this slide. And in this slide, it's talking about where do you get your calories and referred to our diet tracking software. So hopefully all of you have found your software and are ready to roll in monitoring your intake. It takes a little getting used to, and it'll take you a few days, but hopefully by the end of the weekend, you'll be ready to go and counting. Notably, notice the four columns once again: calories, carbs or carbohydrates, fat, and protein. Now, being a biologist, I really think of nutrition from the perspective of what we call the four macromolecules of life. Of course, the big macromolecule that I like to think about a lot is DNA and any of the nucleotides, DNA and RNA. However, however important they are for nutrition, however important they are for genetics, they are not as important when it comes to nutrition. So I want to focus on the other three macromolecules: carbohydrates, fats, and proteins, and learn a little bit about the chemistry that underlies these three molecules. So notice here, I've got nucleic acids down at the bottom, DNA and RNA. We call them macromolecules because each of these molecules is what we call a polymer in chemistry. That is, a polymer is a molecule that is made up of smaller subunits that get linked together to produce a much larger molecule. So we have polymers and monomers. Linking together monomers makes a polymer. When we think about these, we think about carbohydrates first. These are also known as monosaccharides or simple sugars. Of course, polysaccharides are where we link together the monomer, in this case, simple sugars. We link them together to form a polymer. And how they are linked together defines what type of carbohydrate that we have. And I'll show you some examples shortly. Proteins, of course, extremely important for nutrition. Why? Because they provide us with amino acids, and we'll talk about those as well, and the different types of amino acids. Finally, fats and lipids. Obviously, fats and lipids, very important. They have very, very high caloric content, a source of energy for us. And also, if we eat excess energy, the way our body stores it. And we'll talk about those mechanisms of breakdown and storage a little bit later in the course. Fats and lipids, of course, are polymers of fatty acids. And I'll show you what makes up a fatty acid in a little bit. And usually 3 different fatty acids are linked together via a glycerol molecule. And I'll show you what the glycerol molecule looks like as well. But let's take a look at the different types of sugars. Here, of course, is ribose. It is a 5-carbon molecule. If you take a peek, there is an oxygen at the top of the pentose ring. And notice a few things. Here is a hydroxy group, that is oxygen and hydrogen. This is actually the first carbon, then we see at each other point, except for the O, at each other point, we have another carbon. So 1, 2, 3, 4, 5 carbon molecule, and at the very top, an oxygen unit. Notice that for ribose, we have 2 hydroxy groups at the bottom. You don't need to know this in as great detail, but you should know that ribose is a 5 carbon sugar and is shaped as a pentagram. Deoxyribose, which may sound familiar to you. Why? Because you probably know DNA, deoxyribonucleic acid. And in fact, this is the primary sugar found in DNA. And notice it is still a 5 carbon sugar. It is just deoxygenated. That is, it is missing an oxygen. And you can see at carbon 1, 2, 3, 4, it is missing an oxygen group, okay? And just has a simple hydrogen. But we do have other sugars of interest. Here's our buddy glucose. Now notice glucose is a 6 carbon molecule. Again, we start counting from this carbon group in the upper left-hand portion. 1, 2, 3, 4, 5, 6 carbons, again connected by an oxygen group. And notice how in glucose, the hydroxy groups on the lower carbons of the 6-member ring kind of alternate their structure. So that's glucose. This is the molecule, when we talk about metabolism, that your cells use to produce energy. This is used by all cells to produce energy. Here's another one. This is galactose. Galactose is also a 6 carbon. Again, don't worry about the structure, but you should know that glucose and galactose are both 6 carbon monosaccharides. Here's our buddy, and I think that you have all heard of this one, fructose. Fructose is interesting because it is shaped like a pentagram, but notice it has 6 carbons. And again, if we look at the top left, 1, 2, 3, 4, 5, and then the 6th carbon is down here hanging off of the 5th carbon. Sorry. So we can see very, very clearly that fructose is a 6 carbon, 5-member ring, and that's the part you have to remember. Don't worry about the structure itself. I'll never ask you to reproduce it, but you should know it's 6 carbon, 5-member. Of course, where's fructose found? Okay, the clue is FRU, fruit. Most fruits, and in fact, most vegetables, produce fairly high concentration of fructose. So we've got glucose, galactose, fructose. The ribose we don't really worry about too much because they're found in DNA and RNA. But if we were to look at how these are connected to produce more complex sugars, and in fact, here's one that everybody knows and loves, sucrose. Note, sucrose is actually a disaccharide that is made up of 2 monosaccharides, and you should be able to recognize rather quickly that glucose plus fructose equals sucrose, and they're connected again by this oxygen group between them. So if you add sugar to your coffee, or during your baking or at any point in time, what you're really adding is both glucose and fructose, all right, combined together. So what this really means is that sucrose is 50% glucose and 50% fructose, 50-50, okay? Now, there are only really 2 plants that actually make sucrose directly. okay? And those two plants are sugar cane and sugar beets. So those are the two plants that actually make sucrose directly. You can crush them up and extract the sucrose. Makes you wonder for a minute, what is high fructose corn syrup? Well, we already know that sucrose is 50%, right? 50% fructose. How much fructose is in high fructose corn syrup? The answer might surprise you. It's actually 55%. So it's 55% fructose and 45% glucose. Not quite as high as you would imagine. Higher, certainly, than sucrose, but not quite as high as the name might imply. And we'll talk about the impact of high fructose corn syrup on nutrition and health a little bit later on. Here's one of my favorites, maltose. Maltose is actually two glucose molecules hooked together, and you can see that very simply in our little diagram here. Maltose, of course, and you should know this probably is very important to some of you. Malt, anybody drink a malt beverage on occasion? A malt beverage, of course, is beers, ales, White Claws. If you happen to drink White Claws, the alcohol is created from the digestion of malt into ethanol. For me, it's a little bit different. Maltose is one of my favorite because malted grains produce whiskey, and I happen to like Scotch, so this is one of my favorite sugars right here. Again, another economically important disaccharide. Now, when we think about more complex saccharides or starches, we can kind of begin to see that these starches are polymers of glucose molecules. And notice how they're all hooked together. One of the simplest of all starches, amylose. Amylose is just a string of glucose molecules in kind of a boxcar fashion. It's just a simple string of glucoses hooked together, many more than 5, many more than the ones that I'm showing you here. And this is just to give you an illustration. And notice that they all occur in a single line. Amylose is produced by plants. Starchy plants, you'll find them in potatoes, you'll find amylose in rice, you'll find amylose in wheat and flour. And in fact, the first portion of the digestive process, which again we'll talk about in a week or so, occurs in your mouth because your saliva contains amylase. Anytime you hear the ending ASE, it means there's an enzyme. And the function of amylase is to break down amylose. You may recall, perhaps from kindergarten, your teacher may have given you a piece of white bread, probably without the crust, and said, keep chewing, don't swallow. Just keep chewing and chewing and chewing. And as you chewed, what first appeared to be starchy starts getting sweeter and sweeter and sweeter. And the reason is the amylase in your saliva starts to break down the amylose into individual glucose molecules, which your tongue responds to and you taste as sweet. So amylose is an important starch. Very often in the nutritional literature, you'll hear of this as being referred to as a simple starch, meaning it is completely linear. Of course, the other example at the bottom blue box is amylopectin. And notice amylopectin is a branched chain starch. And notice how they are connected. So instead of just a simple boxcar train design of molecules, these have very branched chains. And of course, different enzymes break up the oxygen bond between the different branch chains than would break the oxygen bonding between individual glucose molecules. So it takes a little bit different type of digestion. And this is why when we talk in nutritional terms, that the complex starches or the branch chain starches take longer to digest to break down into individual glucose components, because you have to break down the branches themselves, not just the oxygen bonds in between the glucose molecules. Now, some people have shown over the years that because the linear chain in amylose is easy to break down, begins in your mouth, when you swallow it, you're actually swallowing more glucose. When glucose hits the small intestine, gets absorbed directly into the blood, which causes a very sharp rise in insulin. Whereas if you eat complex starches, like these branch chain starches, again, some of the bonds will be broken, but it is a much lower concentration of glucose, and it takes other enzymes in the small intestine to break down the branches. And so as a result, there's slightly less glucose being absorbed by the bloodstream. Eventually it's all the same, but it is a much slower time scale, at least in the time scale of absorption. And it's a much slower time scale, and therefore you have an attenuated rise in glucose and insulin levels in the blood. Something a lot of people forget, and this is that cellulose is actually a starch. And notice that the only difference between the amylose on the bottom, that's that linear boxcar design of starches, and cellulose in the upper portion of the diagram, notice that it's the shape of the bond that's different. And this beta-acetyl bond cannot be digested by amylose. Therefore, you cannot digest cellulose. You cannot digest cellulose because amylase, I think I said amylose, amylase cannot digest the beta-acetyl bond. So cellulose, if we could break it down, would be a very, very potent source of energy for us. But of course, we can't. There are some animals that are specialized in digesting beta-acetyls, and this is usually accomplished by things like termites, cows, and other ruminants that have a commensal or at least symbiotic relationship with bacteria in their gut, and the bacteria can digest the beta-acetyl bonds. Finally, let's take a quick, or actually not quite finally, almost finally, let's take a look at amino acids. Amino acids are called an amino acid group because you can see on the left, we have an amino group, that's that NH2 to the left, and we have an acid group on the right, that is that COOH group on the right, and in the center is a carbon. Now notice above that central carbon is an R group. So all amino acids have an amino on one side, a carboxy on the other, or an acid on the other, and they differ by the R group. There are 20 naturally occurring amino acids, and everyone has a different R group. Now, what do you need to know? Will I ever ask you to draw an individual amino acid? No. But you should know that an amino acid has an amino group on one side, a carboxy group or an acid group on the other, and a carbon in the middle. And what differentiates them are these different R or residual groups. I have diagrams of all of these amino acids in the next few slides. You should take a peek at them. What you really should know for nutritional purposes are the names of the amino acids. It's not necessary for you to know the structure of every amino acid. However, these amino acids are usually divided from a biochemical perspective in terms of their charges and their hydrophobicities, that is or hydrophilicities, that is their love or fear of water. So they either attract water or repel water. These happen to be hydrophobic, they repel water, but again, not that important. But the names of the amino acids you should know: alanine, isoleucine, leucine, valine. Notice that every amino acid has both a 3-letter code and a single-letter code. It's not necessary for you to know these codes, but they're provided here for informational purposes. Here are the aromatics. Notice that the aromatics each have a 6-member carbon ring attached to them. Their names may sound familiar to you: phenylalanine, tryptophan, and tyrosine. These are important amino acids because not only are they involved in the construction of proteins, but they also may have neurotransmitter activity and certainly are precursors to different neurotransmitters and hormones in your body. Tryptophan, for example, is a precursor to serotonin, tyrosine, a precursor to epinephrine and norepinephrine. So each of these plays a role in the control of the nervous system. Here are some of the polar groups: asparagine, cysteine, glutamine, methionine, serine, threonine. If you take a look at the chemical structure and think way back to when you had chemistry, both cysteine and methionine are the sulfur-containing amino acids. The sulfur-containing amino acids. There's aspartate and glutamate, very similar to asparagine and glutamine, only instead of an additional nitrogen group, they have that acid group at the end. Anytime you hear the ending or suffix ATE on a chemical, it means it's an acid. So aspartate is also known as aspartic acid. Glutamate, also known as glutamic acid. Arginine, histidine, lysine. Glycine is the simplest of amino acids, because if you take a look at the structure, on the left there's an amino group, on the right there's a carboxy group, and the central C, that's it. That's it. No actual R group in glycine. Proline's a cute one. There is both proline and hydroxyproline. Look at the funky shape. The nitrogen is actually encompassed, the amino group is actually encompassed in a ring structure. Proline and hydroxyproline are very important for the formation of cartilage and collagen fibers. And if we get a chance, we'll take a look at those a little bit later on. Ultimately, and again, don't let the chemistry scare you in this slide, ultimately, amino acids are linked together by something called the peptide bond, and you can see that the amino group on one side of one amino acid binds to the carboxy or acid group on the other side of another amino acid, and when water is removed, it creates a very, very powerful bond between the amino acids. This is known as the peptide bond. The interesting thing to me about the peptide bond is that if you were to try to break that bond in the laboratory, it has to be broken in a 6-normal hydrochloric acid solution. That is the most concentrated hydrochloric acid solution that you can produce. at 100 degrees centigrade for 12 hours. That is, and by the way, it can't include oxygen. It's got to be under nitrogen or any non-oxygen gas. So it has to be boiled in concentrated hydrochloric acid for 12 hours. That's how you break the peptide bond in the lab. Again, what makes it interesting to me is your stomach does it, or rather your small intestine does it in enzymes released by the pancreas, which we'll talk about the digestive process again next week. This process is done in a matter of seconds. So with the correct enzymatic treatment, this is done very rapidly, certainly enough time for you to get hungry for lunch again after you eat breakfast. But certainly not in the context of a laboratory. So it's kind of interesting. They are one of the most tough and strong bonds in all of biochemistry. And in fact, if you want an example, spider silk, as I'm sure you know, has very, very high tensile strength, nearly as much as steel, but has a fraction of the weight and volume of steel. So spider silk is tremendously strong, and the reason it's strong is it is a protein and is a string of amino acids connected by the peptide bond, which is a very, very powerful bond. From a nutritional point of view, obviously amino acids are important. These are proteins. Proteins are broken down by your digestive tract into amino acids, most of which are absorbed individually. Again, we'll go over this when we talk about digestion. They are absorbed into the blood and transported throughout the body to serve as building blocks for proteins. Mostly, if you think about where all the protein is in your body, it's in your muscles. It's in your muscles. That's where all your protein is. And in fact, the fluids within your muscles serve as a potent reservoir of free amino acids. And why? Because your muscles are constantly building and breaking down proteins. Now, some amino acids can be synthesized by the cells in your body. Some cannot be synthesized. Those that cannot be synthesized are called essential amino acids. And of course, these essential amino acids are listed in the left-hand column: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Okay, those are essential amino acids. There are some amino acids that are considered to be conditionally essential. Conditionally essential amino acids means that there are certain disease or pathophysiological states in humans. Some are genetic disorders, some are diseases, where these amino acids that are normally synthesized by your cells cannot be synthesized by your cells. One example is in the case of phenylketonuria. In individuals with PKU, cannot synthesize tyrosine. So tyrosine is conditionally essential. The other amino acids, alanine, aspartate, asparagine, glutamate, serine, and cysteine, and in this case, selenocysteine and pyrrolysine, which is not used by humans, all of those are non-essential, meaning your body can synthesize those. This list is different for different organisms. Different organisms have different essential amino acid requirements. So when we think about them, your dog, your cat, okay, don't forget, insects are animals. All animals may have different essential amino acid requirements. These are for humans only. It's always a good idea, just for a moment, to talk a little bit about fatty acids. Fatty acids are very long series of hydrocarbons. because these are just made up simply of carbon and hydrogen. They tend to be hydrophobic. That is, they repel water. Usually, 3 chains of fatty acids are bound together by the green glycerol molecule on the left of the diagram. Notice glycerol has 3 carbons, and in fact, those carbon chains are connected. The glycerol molecule, of course, is hydrophilic. So one of the unique things about fatty acids, and in particular fats, are that one end of the molecule is hydrophilic, that is, it attracts water, and the other end is hydrophobic. These fatty acids are actually the main component of soap, and this is how the soap surrounds the dirt in your clothes. When you have oily dirt, the hydrophobic end surrounds the oil because it's not water, and then creates a little bubble where the glycerol side of the molecule is solubilized in water and creates a little bubble that allows the oil to be removed from your clothes. So that's pretty much how fatty acids work. It also is why fatty acids are a primary component of all your cell membranes. One side, the side facing out toward the cell, the outside of the cell, is actually hydrophilic, whereas the inside of the membrane is hydrophobic. And so your cells are comprised of a lipid bilayer, that is 2 layers. Again, don't worry about the absolute structure, except for one little thing. Notice that of the 3 hydrocarbon chains that I show you, some of them are just repeats of the same carbon molecule, but that bottom one, take a peek at this bottom one here with a double bond between these carbons. Because these hydrogens are not all lined up together, notice that it causes a kink in the shape of the tail. So it almost gives it a little curve here. This is extremely important when you want to tell the difference between saturated and unsaturated fats, and I'm sure you've heard of this. So saturated fats means that all of the carbons in the carbon chain are saturated or filled with the maximum number of hydrogens that can fit on those carbons. Notice that the unsaturated fat below, there's a kink. And so that kink is caused because it does not have the most hydrogen groups that could possibly be carried by those carbons. And in fact, that particular fatty acid with only a single double bond between the 2 carbons is a mono unsaturated fat. So you can imagine if I ask you what a polyunsaturated fat is, which I'm sure you've read on food labels, polyunsaturated means they have more than a single double bond. Of course, when you have a large number of these kinks, because of the spaces that are created by the kinks in the chain, these fats are all liquid at room temperature. So think about what's in your kitchen for a minute. What fats are liquid at room temperature? Canola oil, polyunsaturated. Vegetable oil, certainly unsaturated, but made of a mix of oils that are both mono and unsaturated fats. Olive oil, especially extra virgin olive oil, is an interesting one because it is mostly mono unsaturated fats. So what types are solid? Well, butter, lard, and of course, Crisco. You've also probably heard about trans fatty acids and the effect of having trans fatty acids that are carcinogenic. And trans fatty acids have been banned as foodstuffs. Basically, this was found out thanks to everybody loving their French fries. And most fast food restaurants would reuse the oil in their French fryers. They would clean it, strain it, but then put it back in. And basically, all unsaturated oils, either monounsaturated or polyunsaturated, when heated to 350 degrees Fahrenheit, move from the cis conformation, that is where the hydrogens are on the same side of the carbon double bond, to the trans configuration, that is they're on opposite sides. Again, these are very simple to draw. You just need to know that the trans fatty acids have the hydrogen atoms on either side of the double bond. And all cis configurations, which is how they are normally found, get converted to trans at 350 degrees Fahrenheit. So of course, if you're constantly reusing frying oil, you are constantly reheating the oil to 350 degrees, creating higher and higher concentrations of trans fats. Excuse me. So this put a situation where people, when they ate their French fries, were actually getting very, very high levels of trans fats in their diet. Trans fats were found to be carcinogenic, and so very, very few people, in fact, no one really recycles their oil anymore back into the fryer. The fryers are drained regularly and are sent out to be recycled, usually get put into animal feed or get converted into other useful compounds. And that brings us to the end of the chemistry part of our show. I hope that it was not too technical. I think it's a pretty good overview, and it's not bad to stretch a little bit. I promise next time I'll shave and be nice and clean shaven for you guys, and in the next set. Hope you have a great Memorial Day weekend. Please remember to be safe, stay well, and I'll talk to you. Bye-bye.